Hi everyone, welcome to our online Macromolecules Lab. Just a reminder, my name is Sarah Ward and I am your TA for this course. My email address is sarah.ward@ou.edu. So if you need any help or ever have any questions, again, feel free to email me and I should be able to give you a pretty quick response. So today's lab is macromolecules, identification and analysis. And in this video, I'm going to go over some of the helpful tips that you're going to need to do well in this online lab. So what you should already have completed is listed here on the slide. There is a document called the Macromolecule Lab PDF, and you should have this document printed off of your Canvas. At this point, you should have read all of the pre-lab information that is provided on pages 1 through 3, and also completed the pre-lab questions on pages 3 and 4. If you have not done that, go ahead and pause this video right now and go do that because I'm assuming that you're coming from a place where this is already completed and you're ready to go like you would be if you showed up to an in-person lab. In this lab, what we'll be doing is identifying and describing various types of macromolecules and then trying to decide whether or not they're in certain common foods, and then writing a summary and making a comment on Flipgrid, but we'll get to that in a minute. So starting with macromolecules, one of the most common types are the carbohydrates, which are sugars. They can be monosaccharides, like you see here in the top column, like glucose and fructose, and those are used as cellular food sources. If these sugars are present in a certain type of food, then they will be indicated by Benedict's reagent. That's one of the chemicals we will be using for this lab, and it turns orange or red if there's sugar present. So if you have a tube, add some Benedict's reagent, it turns an orange or a red color, that means there is sugar present, and if it does not turn one of those colors, that means there's not one of these sugars present. So monosaccharides, again, are these common things like glucose and fructose. And then sometimes they end up getting joined together through things like dehydration syntheses to form things that are sucrose. Mono is one of the prefixes that means one, and di means two. So monosaccharides are a simple sugar, and then disaccharides happens whenever two of these simple sugars get linked together. Another type of carbohydrate are your starches. These are polymers of many glucose molecules. Now, a polymer, if you don't already know, means many pieces. So poly means many. Like I just talked about, mono is one, di is two, poly is many. So whenever you see here in the top row where we have various glucose molecules linked together, these make a polymer, and that creates a starch instead of a simple sugar like we had on the previous slide. This is used as structure and energy storage in a lot of plants. So it's a slightly more complicated version of the simple sugars that we use for cellular energy in animals and humans. The presence of starches is indicated by iodine in these tests. So if you add iodine, the tube will turn black if there's starch present, and if it does not turn black, that means there is not starch present. Another category of macromolecules are proteins, which are polymers of amino acids. And again, polymer means many parts. So many of these amino acids linked together makes a single protein or multiple proteins. They have various types of folding structures. You can have highly folded structures that are used for really complex things inside of the cell, or some more simpler folding structures. But the folding pattern of proteins is really, really important to the function. So proteins are used for various things. They are in things like enzymes, which do various tasks within cells, and also um, we are looking at this in one of our other labs. So if you are in B group, then next week you will be looking at enzymes in person. And if you're in A group, then you did this last week before you got to this online lab. But our potato experiment in person is one of these enzyme activities that we were looking at the function of. They also can be used for things like cell structure or transport, where they actually make up the backbone of cells, so to speak. Obviously, cells themselves don't have bones, but they kind of give some support and structure to the cells and also moves various things around within the cell. So all of these are very important to life as we know it. The presence of protein is indicated by the Bure reagent, which turns purple whenever it gets added to a tube that has proteins in it. So that's what you need to be looking for. If the Bure reagent turns purple, that means there is protein present. And the last group of macromolecules that we'll be talking about today are glycerol and fatty acids, which are both types of lipids. So lipids are a little bit funny. They have things called hydrophilic heads and hydrophobic tails. So you can see here, we have the various things going on. Here are our hydrophilic heads, and here are the hydrophobic tails. So let's break down these words a bit to help us understand what they mean. Hydro means water, philic means loving, and phobic means fearing. So if you have a hydrophilic head, that means it's going to be attracted to water, and a hydrophobic tail means it's going to be repelled by water. So what these end up doing is they form what's called a lipid bilayer. Well, all of these heads link up together, and then there'd be another row of heads down here that link up together, and the tails would be in the middle. That's because the heads are attracted to water inside and outside the cell, while the tails are repulsed by those. So usually these tails will do whatever it takes to get away from water. Again, that is found inside or outside of the cell. So usually they come in bilayers so that these tails can hide from the water. They are used for things like energy storage and memories, is what I was just talking about with the bilayer. But they're also present in various things such as butter, oil, that kind of thing. So if you've ever, let's say, washed dishes, for example, and you had a really greasy pan from making bacon or something like that, if you've noticed, whenever you put water in that pan, you'll have these little droplets of oil float up to the top, right? They don't mix together. So, like the old saying, water and oil don't mix, right? That's because of this hydrophobic tail sort of situation that goes on. So if they can't form a bilayer like they do for membranes, then they'll just like huddle together in a single droplet, which will allow them to avoid the water. The presence of lipids is indicated by Sudan 4 reagent, which turns bright red if there's lipids present in the test subject. So what we'll be doing in the lab is trying to determine which of these macromolecules are common in various types of food. So we have prepared for you a video where somebody actually performed this experiment, and you'll be able to see these different tubes turn colors. And based on that, whether or not there's a positive or negative reaction to these various tests, you should be able to tell which macromolecule is present in which food type. So, setting up the experiment, we need to have control groups. Now, let's talk a second about the purpose of controls. Controls are to ensure that your experiment is working properly. So, we have what's called positive controls, and we also have what's called negative controls. So a positive control contains only the molecule of interest, so we know for a fact we should get a positive control if a positive result if everything's working correctly. So for example, if we want to test our reagent for protein to make sure it's working properly, then we will place it directly on something that we know for a fact is protein. And then if it gives us the positive reaction that we're looking for, the color change, then we know that our chemical's working well and that things are um, working as we expect. A negative control should not contain any of the molecule and also kind of functions in the same way. So let's say we drop some of that reagent that just gave us a positive reaction for protein. If we drop it in water, it should not give us a positive reaction. So having a positive control and a negative control together can ensure that your chemicals are nice and working properly like they should be, and that the reaction you're seeing is actually based on what's happening and isn't just some strange thing like having an old chemical or one that's been too hot or too cold or whatever that might affect the activity of these particular chemicals that we'll be using. Our experimental groups are going to include hot dogs, tofu, an energy drink, and milk. And we're going to be testing each one of these four things to see if it contains a simple sugar, if it contains a starch, if it contains a protein, or if it contains a lipid. So our dependent variable is going to be the type of macromolecule present, because remember the dependent variable is what we are actually measuring. The independent variable is going to be the type of food tested. If you remember the little trick I gave you last week, the independent variable, the I indicates I am changing this. So I am changing whether we're testing hot dogs, tofu, energy drinks, or milk. And so that is our independent variable is the type of food. And the dependent variable is going to be the reactions that we get, whether they're positive or negative. So remember when you're writing your hypotheses for this experiment that it should be a causal explanation. The goal of having a causal explanation as the hypothesis is that it can be tested. It doesn't necessarily have to be right. There are many, many cases of science. I could give you some from my own research, where I came up with a hypothesis, made an educated guess, and then turned out to be exactly wrong. So, for example, one of your hypotheses could be something like Red Bull turns back in the presence of iodine because it contains starch. Then, when you go to test it, if it turns back, then maybe it does contain starch, and you can support that hypothesis. But if it does not turn back, then you can say that the hypothesis can be rejected. So, as a summary, these causal explanations should be a best guess. You should try to be serious with them, but it's okay if your hypothesis is wrong. But right now, I want you to go ahead and write a hypothesis for each of the four foods that are being tested in this lab. These will be included in your lab summary, so it's important to put a little bit of thought into this and try to come up with good hypotheses. Again, remember, they need to be specific enough to be testable, such as this Red Bull turns black because of the presence of iodine because it contains starch. This is something we can test. If it turns black, great, it has starch. If it doesn't, then it doesn't have starch. So make a hypothesis that is specific and testable for each of the four food groups. You should go ahead and pause this video now to do just that, because this is the stage where you should be writing these hypotheses before you go on any further. So pause for a few minutes, write down a hypothesis for each of the four food groups being tested. Okay, great. By now you should have your hypotheses written down. So I'm going to go through as if you were in lab doing this experiment yourself. The very first step would be to label the test tubes, like, again, we are doing for the enzyme activity that you either have already done or are getting ready to do next week. You'll use these four different reagents to identify specific macromolecules. There's iodine, Sudan, Bure, and Benedict's. Reagents can stain and irritate your skin and eyes. So in person, when we do this experiment, we would always wear goggles and gloves when handling the reagents and the test tubes, just to ensure that you didn't end up getting any of it on you, to be super safe. We would then use the markings on droppers to transfer 2 milliliters of each test solution to each tube, and then add the amount of reagent called for in the protocol. So I believe it's in your lab background how various tests need various amounts added to each one, whether it's a few drops or a milliliter, however much it is, you would follow the protocol to add the correct amount of reagents to 2 milliliters of each of our test subjects. So the first thing we would do if you were in person would be to plug in, turn on, and adjust the temperature on the hot plate because it takes about 20 minutes or so for the water to boil that you would need for this Benedict's test. But again, since we're online, you don't need to worry about that. Just be aware that it does take a while for the Benedict's test. So, whenever you do get water in and out of hot test tubes, make sure you handle those with the tongs. This goes for the entire semester. It's really easy to kind of forget that they're really hot because you just get kind of excited, you want to pull it out. But the entire glass does heat up, and so it will burn you, and it is important to use tongs. After we do each lab, you need to make sure that you clean up after yourself and get things put away for the next lab section. So, to clean test tubes, you use soap here in the bottle. You just squirt a little bit in until it just fills up that rounded end, as you can see in the middle picture. And then you use a small test tube brush, like on the right, to scrub it and then rinse it out. At the very end, once all of your test tubes are clean, you give each test tube a final rinse with deionized water in the sink spout that has the white nozzle. And then you turn your test tubes upside down in the rack and place it on the absorbent pad just to get all of the water as it's drying. Then at the end, you need to write this experiment summary and comment with a flip grid. But before you do this, there is a video on your canvas where these experiments have been done and you can just write down the observations. So again, you should have your background printed out. So as you're watching this video of this experiment being done, you can write down your observations of which tests were positive, which ones were negative. And then using that data is what you use to write the experiment summary. And then you'll also need to make a Flipgrid video, which we'll talk about in just a minute. So here's just kind of a funny comic that I thought I would throw in because we all know the struggle of trying to write a summary. But this shouldn't be too hard for you. Just try to be straightforward, follow the instructions. To display your data, some tips. When you have finished collecting your data, you should summarize the data you recorded in one table to use for your lab summary. So the table should include enough information about which molecules are present in each food sample to figure out. So your independent variable should be in the leftmost column, like in table 2 in the lab background. And you should place the dependent variable across the top row. And then in the single graph, you should be able to summarize each of the 4 different food groups that you used to say whether they were positive or negative for each one of the macromolecules. Go ahead and feel free to back up to the side where I was talking about the independent variables if you need some help trying to figure out how to set up this graph. But you can refer to that background in table 2 to help you as well. Some tips to help you be successful with this assignment is to review the How to Write a Lab Summary document in the Lab Resources module on your lab canvas, and or to review the assignment rubric before writing. So whenever I go to grade these, I literally go through the rubric and click whether or not you follow these instructions. So if you follow the rubric really closely, you'll be much likelier to get an A than if you just kind of go off and do your own thing. So I would recommend you to take a look at that rubric and also take a look at the How to Write a Lab Summary document to give you some tips. This lab summary is uploaded to Canvas by noon of the Friday that you're doing this assignment. So B group, you're doing it this first week. This needs to be uploaded by this Friday. It will probably show up that it's open for an extra week because A group is going to be doing this the following week while you're in person. But your lab summary is due by noon of the week that you do this. So make sure you don't turn this in late because I want to be able to give you credit for it. And remember, late work is not accepted, so make sure you get this in by noon on Friday of the week that you're assigned to do the online lab. And then finally, you have a Flipgrid post that is also due by noon on Friday, and you should address the following. It should be a about a minute long. I'm not going to be a huge stickler about that, so if it's 45 seconds, fine, whatever. But try to make it about a minute. And in the video, you need to say your name. You need to give a brief summary of the experiment and the conclusions that you came up with. Just give what the most challenging part of the activity was, whether it was picking an Excel or seeing the colors clearly, whatever it is that you found most difficult about this lab. And then also something new that you learned as a result of this activity. Probably something about macromolecules, but it could be something about lab technique, whatever you think. So make sure that at this point you should go watch the video that is uploaded on Canvas. You should write your lab summary, get that turned in by noon on Friday of the week that you're doing this. And then you also need to do a 1-minute Flipgrid video. And there is a link to this on Canvas as well. So look for your lab section with my last name also associated with that. And you follow that. You will need to use your OU email address to be able to access that. I set it up so that you can make accounts with an ou.edu email address to access our Flipgrid posts. So it probably won't let you do it with Gmail accounts or anything like that. So just use your OU email and write your lab summary and then the Flipgrid video. And I'm really looking forward to seeing what you guys turn in. So please let me know if you have any questions, concerns, comments, and I will try to help you as best I can. But good luck with this. I'm sure you will all do fantastic.